So what is a cell? It is considered the building block of life. All living things are made up of at least one cell, and all those chemical reactions take place inside the cell, performed by cell components. It is the smallest level of organization we can go and still be considered alive. Through our studies of the cell, we've been able to discern that there are two distinct cell types, which ultimately have given rise to three scientific domains: archaea, bacteria, and eukaryote. The simple prokaryotic cell consists of two domains, archaea and eubacteria. The eukaryotic cell encompasses all complex life, protist parasites, fungi, animals, and plants. For now, let's focus on the big distinctions between prokaryotes and eukaryotes. The prokaryotic cell is always single-celled and gives rise to simple single-celled organisms. When I say simple, I mean that cell has no nucleus. Its DNA is free-floating in this soupy matrix that is the cytoplasm. There are no organelles or subcomponents that do work within the cell, which means everything that this organism uses is osmosis or diffused in from the outside world. Prokaryotes evolved alone on our planet for 1.5 billion years. They are the earliest living organisms, and therefore they had the most time to reproduce, making them the most abundant and adapted organisms on our planet. They live everywhere we are and everywhere we aren't, including places too salty, too basic, too acidic, too hot, too cold to sustain any other life. The eukaryotic cell can describe life that is single-celled or multi-celled organisms. But the big distinction here is the eukaryotic cell is complex. The DNA is sequestered in a nucleus, safely stored away from the rest of the cell components. There are many membrane-bound organelles that do work within the cell. If you look at the pictures, we can see an animal cell and a plant cell, and it's color coordinated to show the similar organelles in both spaces. But these cells are full and bursting to the brim with components in comparison to the prokaryotic cell we saw previously. Because they have these subcompartments that can do work, they don't rely wholly upon osmosis and diffusion for their nutritional needs. They can sometimes make their own. And so this allowed these cells to get slightly larger, both to accommodate the extra organelles, but also because of the functionality and efficiency that organelles provide them. All cells will have a plasma membrane. All cells will have a cytosol or cytoplasm. These are interchangeable words that refer to the semi-fluid liquid substance inside. All cells must have ribosomes, and all cells will have genetic information. So let's talk about these basic components first. The plasma membrane. It is a selective barrier that allows the cell to keep separate and distinct from the outside world, but also allow things to pass through to obtain nutrients and building blocks for future reactions and growth. The plasma membrane is built of a double layer of phospholipids, one facing the outside world, one facing the inside of the cell. We describe these phospholipids as components exhibiting a hydrophilic, water-loving head and a hydrophobic, water-fearing or water-avoiding tail. And so this orientation works because the inside of the cell is the cytosol, which is mostly water, and because the outside of the cell, the world, is also largely water. And so this creates a little barrier in which in versus out distinct sides can be made. Sometimes called a fluid mosaic model because it is a mosaic of multiple pieces. There's the phospholipids, there's cholesterol, but there's also transporters, enzymes, surface receptors, adhesion molecules, anchoring proteins. The idea here is to recognize that the plasma membrane is more than just a wall or a barrier. It moves things in and out of the cell. It can do chemical reactions right at the surface of the cell or within the cell. It can communicate between cells. It can attach cells together to form sheets, and it can also anchor a cell in place to build three-dimensional structures and models. The plasma membrane is a very robust and integral component for the cell. The cytosol, or cytoplasm, is the intracellular fluid that exists within the cells. It's largely gelatinous. The idea here is it's mostly water, so it can dissolve molecules and help carry them across the cell. suspend the other elements like the ribosomes and the genetic content within the cell, but it also exerts a little bit of surface tension and force against the plasma membrane to give the cell its shape and structure. The number of chromosomes will vary by organism, but in all cases, this is the genetic blueprint that describes what the cell can do, its structure and functionality. And finally, all cells must have ribosomes. Ribosomes are the translators. They are able to read the genetic information and translate that into proteins, the doers of the cell. Let's look at the complexity that eukaryotic cells have over their prokaryotic counterparts. Eukaryotes have a nucleus. This is a double-layered compartment within the cell that contains chromosomes, that sequesters the genetic material away from everything else in the cell. This provides separation and protection to the genetic content. Inside the nucleus is a nucleolus. This is where ribosomes are made. Remember that ribosomes are required for all things, all cells, and so eukaryotic organelles are able to make more ribosomes and therefore outproduce prokaryotes. Right next to the nucleus is the endoplasmic reticulum. It has two distinct regions, a smooth section and a rough section. The rough section gets its name because it is rough or bumpy, and these are in reference to the physical ribosomes attached to the endoplasmic reticulum. The smooth area lacks ribosomes. Depending upon whether we're talking about the smooth or the rough ER, the endoplasmic reticulum will have different functionalities because remember, form dictates function. You don't have to know all the things the endoplasmic reticulum can do, but be aware of its primary role. Make lipid, detox, move stuff. So here we can see the end of the nuclear envelope in purple, followed by the rough ER, giving way to a smooth ER. If we look at the illustrated version of these organelles compared to the scanning electromicrograph of these cells, we can actually see the ribosomes in the folds and the globular smoothness of the smooth ER as separate and distinct areas. Notice the transitional ER, the smooth ER, is blebbing off vesicles, small lipid compartments, and these will move to the Golgi body or the Golgi apparatus. The Golgi apparatus is constantly in flux, pinching off bits and getting smaller, and receiving bits and getting larger. The Golgi apparatus modifies things that are received from the endoplasmic reticulum, sorts and packages the cells for long-term deliveries across the cell, or even to neighboring cells. And in the Golgi apparatus, the small raw materials that come in can be sorted in such a way to build the requisite macromolecules in-house. Lysosomes are specialized compartments that contain digestive enzymes. They are used as recycling centers to break down old proteins, fats, polysaccharides, to break down macromolecules back into their small monomer forms. Phagocytes refer to cells that eat things, but the process known as phagocytosis describes how a cell can absorb and engulf food from the outside world. So phagocytosis is the acquisition of outside material, and these phagocytes will fuse with lysosomes and recycle raw material down. Vacuoles are maintenance compartments or storage compartments within a cell that typically hold sources such as water or salts. Everyone's favorite organelle is the mitochondria, the powerhouse of the cell. This is where energy is made. Plants have chloroplasts, which also make energy, but they are the site of photosynthesis, a different energy reaction. Another cell component is the peroxisome. These are similar to lysosomes, but these are specialized toxin digesters that find hydrogen peroxide in the cell and reconvert it back into water. They are basically de-acidifying, detoxifying the cell. They also have a role in breaking down fatty acids. The eukaryotic cells are larger, so they need some cytoskeletal networks to help provide structural integrity and support, help maintain the shape. The cytoskeleton also becomes essentially roads or avenues for transporting molecules across the cell. There are essentially three types of filaments. So microfilaments, the thinnest, are composed of only two thin strands of actin subunit, and they generate a gentle force against the plasma membrane barrier. Their whole function is to just maintain structural shape. Intermediate filaments are composed of 8 sheets of 4 actin-thick filaments, and so they are much stronger, and they allow the cell to anchor and hold on to organelles. Microtubules are even larger still. They are formed by 9 triplets around 2 smaller microtubules, and these components are used during cell division to rein in the chromosomes, to pull something of large mass, not just hold it in place, but move it. If we look outside the cell, we can see that there are cilia and flagella. Now, flagella can exist on prokaryotes and eukaryotes, but cilia is reserved for the eukaryotic structures. These types of filaments differ in their abundance on a cell, but also the type of motion they create. Flagella have a propeller-like motion that drives things forward, and cilia ebb and flow. They generate more of a wave component. Also on the outside of cells, you might see a wall to provide additional support, regulation, a structure to adhere to bind to, and there may also be junctions or gaps in this wall to enable the passage of molecules and communication. Again, we see form dictates function here. Tight junctions seal neighboring cells side by side together. This creates a waterproof layer that prevents leakage between those individual cells. This is how we generate tissues. Adherens junctions have thin actin attachments. Desmosomes have thicker actin adjustments, and they allow these cells to bind and adhere. Gap junctions have holes, which allows for the passage of material. And hemidesmosomes, think like a hemi-engine. These are really strong intermediate filaments. They allow the tissues to attach to the base, the basal lamina, to build upward and create three-dimensional structure. The endosymbiont theory, or the observation of endosymbiosis, describes how an early ancestor of the eukaryotic cell was once living side by side, 2 prokaryotic cells living side by side, single-celled and simple, but one engulfed the other. And instead of digesting down its raw materials, breaking it down, the internal prokaryotic cell persisted. It was able to coexist and survive inside the other cell, and ultimately, they merged into a single organism. We know for a fact that the mitochondria and chloroplasts came about this way. Our proofs are that these are the only 2 organelles that are double membraned. One membrane is the original bacterials, and the second membrane is what they absorbed when they entered into the larger prokaryote. Also, these 2 organelles contain their own free ribosomes and their own DNA. Their ribosomes are also smaller than those ribosomes that exist in the cytoplasm. They also behave independently. We say that they are autonomous. And when we make more mitochondria or chloroplasts, their reproductive mechanism of copying is very similar to that which we see in binary fission amongst bacteria.